Welcome to Chapter 17, Gene Expression, from Gene to Protein. The information content of genes is in the specific sequences of nucleotides in the DNA. The DNA inherited by an organism leads to specific traits by dictating the synthesis of proteins. Proteins are the links between genotype and phenotype. Gene expression is a process by which DNA directs protein synthesis, or DNA is used to make proteins. This includes two stages, transcription and translation. So here we see a picture of albino donkeys, and they are going to be albino because they lack the pigment melanin, which makes them white-furred. This is a result of a single faulty gene, a mutation. How was the fundamental relationship between genes and proteins discovered? In 1902, British physician Archibald Garrod first suggested that genes dictate phenotypes through enzymes that catalyze specific chemical reactions. He thought symptoms of an inherited disease reflect an inability to synthesize a certain enzyme. Cells synthesize and degrade molecules in a series of steps, a metabolic pathway. So to the right, we see a metabolic pathway, which consists of more than one chemical reaction. We have our first chemical reaction, where we convert the precursor into ornithine. We have our second reaction, where we convert ornithine into citrulline. And we have our third reaction, where we convert citrulline into arginine. As you can see, each chemical reaction is going to be catalyzed by an enzyme. Enzyme A catalyzing the first reaction, enzyme B catalyzing the second reaction, and enzyme C catalyzing the third reaction. Our final product is going to be arginine, which is an amino acid. George Beadle and Edward Tatum exposed bread mold to X-rays, creating mutants that were unable to survive on minimal media. X-rays are examples of mutagens, which are going to cause mutations in the DNA. Their colleagues, Adrian Serb and Norman Horowitz, identified three classes of arginine deficient mutants. Each lacked a different enzyme necessary for synthesizing arginine. The results of the experiments provided support for the one gene, one enzyme hypothesis. The hypothesis states that the function of a gene is to dictate production of a specific enzyme. So here we see a diagram of Beadle and Tatum's experimental approach. Step 1, the neurospora cells, or the bread mold cells, are placed in a medium inside of a petri dish, and the cells are going to be exposed to X-rays. The surviving cells are going to form colonies as they divide and grow, and some of these cells are going to be put in different vials. Some of these cells are going to be put into a vial with only minimal nutrients, and other cells are going to be placed in minimal medium, however with additional amino acids. So the first test tube here has a minimal medium plus valine, second minimal medium plus lysine, third minimal medium plus arginine. The control, the positive control, is the wild type cells. The wild type cells are the cells that were not exposed to the X-rays that are growing on minimal medium. So normally the cells are able to grow in minimal medium, but the X-ray exposed mutants are not able to grow in the minimal medium. And as you notice right here, the X-ray exposed mutants are only able to survive in the minimal medium with the arginine, which is the essential amino acid for the survival of the cells. So here we see a chart of the results, and as you can see, If there was growth in the medium, we see that dark yellow area on the top of the medium. If there is no growth, we don't see that dark yellow area or mass. So we have three different types of mutants. We have the class 1 mutants, class 2 mutants, and class 3 mutants. The class 1 mutants have a mutation in gene A, which codes for enzyme A. So if there is a mutation in gene A, enzyme A is deactivated, and the precursor is unable to be converted into the ornithine. Now, where is the precursor found? The precursor is going to be found in the minimal medium. So basically, all of the tubes have the minimal medium. So MM stands for minimal medium. The control is the minimal medium, but we have three other test tubes with either ornithine, citrulline, or arginine. Okay. So for the class 2 mutants, we have a mutation in gene B, which codes for enzyme B. But if we have a mutation in gene B, enzyme B is deactivated, and we are unable to convert ornithine into citrulline, which means we cannot get arginine in the end. In order for us to have growth, we need arginine. We need arginine. Okay. The class 3 mutants finally have a mutation in gene C, which deactivates enzyme C, and we are unable to convert citrulline into arginine. The worst case scenario is class 3, because even though we have enzyme A or enzyme B, we can only get as far as citrulline, and we cannot get arginine at the end because of the inactivation of enzyme C. However, when we look at class 1 and class 2 mutants, even if we deactivate enzyme A, if we add ornithine or citrulline to our test tube, we can get arginine in the end. For class 2 mutants, even though we deactivate enzyme B, if we add citrulline to our test tube, then citrulline can be converted into arginine. So if you want to visualize the results, we have a table up here. And as expected, the wild type, because it has no mutations in any of the genes, is going to produce growth in all 4 test tube scenarios. And this is because we are able to produce arginine. If you look at the class 1 mutants, you cannot grow class 1 mutants in the minimal medium with the precursor. And that is because we do not have enzyme A working. The precursor cannot be converted into ornithine. However, if we add ornithine into the minimal medium, we see that we have growth for the class 1 mutants, because ornithine can be converted into citrulline with the help of enzyme B. Let's skip to class 3 mutants. So for class 3 mutants, we do not have growth in minimal medium plus ornithine, or minimal medium plus citrulline. And this is because we have the deactivation of enzyme C. Even if we had citrulline, we would not be able to convert citrulline into arginine in the end. So this experiment proved that one gene is going to code for one enzyme. Gene A for enzyme A, gene B for enzyme B, gene C for enzyme C. And remember, students, that enzymes are made up of proteins. So each gene is going to code for a protein. So again, we have the control tube right here, where the wild type cells are going to be in, the non-mutated cells, and they're able to grow. But we have our other control, which is the negative control with the mutant cells that are not able to grow in the minimal medium due to the mutation in enzyme A. So again, we see the same chart. Not all proteins are enzymes. So researchers later revised the hypothesis one gene, one protein to one gene, one polypeptide. Many proteins are composed of several polypeptides. A protein can be composed of several polypeptides, or a protein can be composed of just one polypeptide, each of which has its own gene. It is common to refer to gene products as proteins rather than polypeptides. Next, we will discuss transcription and translation, the two steps in gene expression. RNA is the bridge between genes and the proteins for which they code. Transcription is the synthesis of RNA using information in DNA as a template. Transcription produces messenger RNA, also called mRNA. Translation is the next step. This is the synthesis of a polypeptide using information in the mRNA. Ribosomes are the protein factory where translation is going to take place. In prokaryotes, translation of mRNA can begin before transcription has finished. So pretty much they can happen simultaneously. In a eukaryotic cell, however, transcription and translation cannot occur simultaneously because eukaryotic cells have a nucleus. And the nuclear envelope of the nucleus separates transcription from translation. Before the RNA can exit the nucleus and enter the cytoplasm, the mRNA transcripts must be modified through a process called RNA processing to yield the finished mature mRNA. So here we compare the bacterial cell and the eukaryotic cell, and immediately you can see that the bacterial cell does not have a nucleus, and the eukaryotic cell has a nucleus where transcription is going to occur in, as well as RNA processing, and translation occurs in the cytoplasm. In bacterial cells, or prokaryotes, transcription and translation both occur in the cytoplasm. A primary transcript is the initial RNA transcript from any gene prior to processing. The central dogma is the concept that cells are governed by a cellular chain of command, or also you can call this the flow of genetic information. We start with DNA, and DNA is used to synthesize RNA, and RNA is used to synthesize a protein, also called a polypeptide. So how are the instructions for assembling amino acids into proteins encoded into DNA? Remember, amino acids are going to be the building blocks of proteins. Amino acids are also called the monomers of proteins, the proteins being the polymers. There are 20 amino acid building blocks, but there are only 4 nucleotide bases in DNA, A, G, C, and T. How many nucleotides correspond to an amino acid? So right off the bat, it's going to be more than 1. The flow of information from gene to protein is based on a triplet code, a series of non-overlapping 3 nucleotide words. So basically this means that DNA is going to be read in groups of 3, called a triplet. The words of a gene are transcribed into complementary non-overlapping 3 nucleotide words of mRNA, and that is through the process of transcription. These words are then translated into a chain of amino acids, forming a polypeptide or protein. So here we see our DNA at the top, and the DNA is going to have sections called genes. And remember, a gene is going to be important because genes code for a protein. So when we take a look at a gene right here, gene 3 in this case, the DNA or the gene is going to be transcribed into mRNA. mRNA is going to be transcribed from only one strand of the double-stranded DNA, which is going to be the template strand. The template strand is always going to start at the 3' carbon. Lastly, after transcription, we reach translation, where the mRNA is going to be used to synthesize a protein, and the protein is going to be made up of building blocks called amino acids. And we see the different types of amino acids in this amino acid chain or protein chain. And as we saw earlier, there are 20 different types of amino acids. But notice right here, each amino acid is going to be coded by a group of 3 bases on the mRNA called a codon. And this is why we read DNA in the beginning in 3s and eventually RNA in 3s. So in DNA, the groups of 3 bases are called DNA triplets. In RNA, it's going to be called codons. One of the 2 DNA strands, the template strand, which is always going to start at the 3' carbon and then go to the 5' carbon, provides a template for ordering the sequence of complementary nucleotides in an RNA transcript. The template strand is always the same strand for a given gene. The strand used as a template is determined by the orientation of the enzyme that transcribes the gene. This in turn depends on the DNA sequences associated with the gene. During translation, the mRNA base triplets called codons are read in the 5' to 3' direction. The non-template strand is called the coding strand, and the template strand is called the non-coding strand. And this is because the nucleotides of this strand are identical to the codons, except that T is present in the DNA in place of U in the RNA. Each codon specifies the amino acid, 1 of 20, to be placed at the corresponding position along a polypeptide. All 64 codons were deciphered in the early 1960s. Of the 64 triplets, 61 of those codons are going to code for amino acids, but the last 3 are going to code for stop signals to end translation. The genetic code is redundant. More than one codon may specify a particular amino acid, but not ambiguous. Every codon is going to specify a certain amino acid. Codons must read in the correct reading frame in order for the specified polypeptide to be produced. The reading frame is referred to as the correct grouping. This is a codon table for mRNA used to find the amino acid sequence for the protein. For example, if I had the mRNA sequence AUG UUU, I would have to start by grouping the mRNA into 3 nucleotides. So we have 2 codons in this case. So if I were to find the amino acid that corresponds to the first codon, we will look at the left column first. So the first mRNA is going to be A. So you look for A in the left column. And the second letter is going to be U. So we look at the top column. After we do that, we just cross them together, and wherever they meet, we are able to find our 3-letter codon, AUG. And to the right of the codon, we have the name of the amino acid, which is going to be MET, which stands for methionine. So we will put MET as our amino acid and move on to the next codon, UUU. So the same rules apply. We look for the first letter, which is U, on the left column. The second letter is also U, so we go to the top and we cross them together and we find UUU in this box. And UUU is going to code for a amino acid called phenylalanine, PHE. And so we have our resulting amino acid sequence, which is going to make up our protein. So here at the top, it's our mRNA, and at the bottom right here, we have our amino acid sequence. And we know that the amino acids are the building blocks of protein. So we can also call it our protein or polypeptide sequence. The genetic code is nearly universal, shared by the simplest bacteria and the most complex animals. Genes can be transcribed and translated after being transplanted from one species to another. For example, we can take the gene from a firefly that allows fireflies to glow through their bioluminescence, and we can take that firefly gene and incorporate it into a tobacco plant, allowing the tobacco plant to express that gene and therefore also express bioluminescence. Another example is a pig expressing a jellyfish gene that was incorporated into the pig from a jellyfish, which allows the pig also to express bioluminescence. Transcription is the first stage of gene expression. As we saw earlier, transcription is the use of DNA as a template to get a pre-mRNA. And mRNA stands for messenger RNA. RNA synthesis in transcription is catalyzed by RNA polymerase, which pries the DNA strands apart and joins together the RNA nucleotides. The RNA is complementary to the DNA template strand. And remember, the template strand is always going to start from the 3' and end at the 5'. RNA polymerase does not need any primer, like we saw in DNA replication. RNA synthesis follows the same base pairing rules as DNA, except that uracil, U, substitutes for thymine, T. The stages of transcription are initiation, elongation, and termination. Initiation is when transcription is going to start. Elongation is when we elongate the RNA transcript, and termination is when transcription stops and we produce the completed RNA transcript. So let's take a look at this video of transcription.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
The DNA sequence where RNA polymerase attaches is called the promoter, as we saw in the video in our previous slide. In bacteria, the sequence signaling the end of transcription is called the terminator. So again, RNA polymerase is going to first attach to the promoter, and transcription is going to end when RNA polymerase reaches the terminator sequence in DNA. The stretch of DNA that is transcribed is called a transcription unit. So let's take a look at initiation, elongation, and termination in transcription. So during initiation, the promoters signal the transcription start point and usually extend several dozen nucleotide pairs upstream of the start point. Transcription factors are very important in mediating the binding of RNA polymerase and the initiation of transcription. Transcription factors are basically going to attach to the promoter and other areas of the DNA and allow RNA polymerase to bind to the promoter. The completed assembly of transcription factors plus RNA polymerase II is called the transcription initiation complex. So the transcription factors plus the RNA polymerase attached to the promoter is called the transcription initiation complex. The promoter is going to be made up of several T's and A's, and we call this the Tata box. And the Tata box is crucial in forming the initiation complex in eukaryotes. And another important fact about the Tata box is that it's mainly made up of T's and A's. This is important because the hydrogen bonding between T's and A's are a lot weaker than G's and C's because we only have 2 hydrogen bonds between A's and T's. This is going to help the RNA polymerase to separate the DNA and start creating the mRNA transcript. So here we have the promoter region. It's going to be made up of a bunch of tatas or T's and A's. And we see the transcription unit, which is going to be all of the genes that are going to be transcribed into an mRNA, and we see the mRNA right here. Notice how we're only transcribing one of the 2 DNA strands. So the DNA strand that starts at a 3 is called the template strand. So in this case, it's the bottom one. So we start at 3, but notice how the RNA transcript is being made in the 5' to 3' direction. Okay. So here again, we see the Tata box in the promoter. We see the template strand that starts with 3', and the non-template strand at the top that starts with the 5'. The binding of the transcription factors is going to allow RNA polymerase to come and attach to the promoter more easily. This is going to form the transcription initiation complex, which is going to drive the formation of the RNA transcript during transcription. And so the RNA transcript is going to keep on getting longer but that happens in the next step of transcription, which is elongation. So in elongation, the RNA polymerase is going to continually move along the template strand, reading it and creating the RNA transcript. It untwists the double helix of the DNA, 10 to 20 bases at a time, and transcription progresses at a rate of 40 nucleotides per second in eukaryotes. A gene can be transcribed simultaneously by several RNA polymerases. Nucleotides are added to the 3' end of the growing RNA molecule. And we remember that the 3' end is going to be part of the template strand of DNA. So here we see the elongation of our mRNA as it goes from the 5' to 3' direction. And we are only reading the template strand, which starts at 3. The final step of transcription is termination. The mechanisms of termination are different in bacteria and eukaryotes, but the end result is the same. Termination in transcription is going to terminate or end transcription. In bacteria, the polymerase stops transcription at the end of the terminator, and the mRNA can be translated without further modification. In eukaryotes, RNA polymerase II transcribes the polyadenylation signal sequence. The RNA transcript is released 10 to 35 nucleotides past this polyadenylation sequence. So what happens after transcription in eukaryotic cells? So there's going to be a step between transcription and translation. This is called RNA processing. RNA processing happens inside of the nucleus, where we are going to process the pre-mRNA and turn it into a mature mRNA. This processing is important before we exit from the nucleus into the cytoplasm. The processing is necessary. One reason it's necessary is because it helps the mRNA to survive in the cytoplasm environment. During RNA processing, both ends of the primary transcripts are altered. Also, in most cases, certain interior sections of the molecule are cut out and the remaining parts are spliced together. Spliced, in this case, means glued together. So let's take a look at the first alteration or processing that we see during RNA processing. This is the addition of a 5' cap and a poly A tail. The 5' end is going to receive the 5' cap, and the 3' end of the pre-mRNA receives the poly A tail. Again, these modifications share several functions. They seem to facilitate the export of mRNA to the cytoplasm from the nucleus, and they also protect the mRNA from hydrolytic enzymes in the cytoplasm, allowing the mRNA to survive better in the cytoplasm. They also help ribosomes attach to the 5' end of the mRNA. So here we see the poly A tail and the 5' cap, the 5' cap here, and the poly A tail here, attached to the 5' and 3' ends of the pre-mRNA. So the last thing that needs to happen in RNA processing is cutting out certain sequences and gluing together the leftover sequences. The sequences or regions of mRNA that we cut out are going to be called the introns. The introns are called the non-coding regions, which means that the introns are not able to code for a protein. After the introns are cut out, the exons are going to be glued together or spliced together. And the exons remain because exons are coding regions, meaning exons code for proteins. The whole process of RNA splicing in RNA processing removes the introns and joins or splices together the exons, creating a mature mRNA with a continuous coding sequence. So here we see our pre-mRNA with the two ends attached to it, but we need to cut out the introns, which do not code for a protein. After cutting them out, the exons, which are red, are going to be spliced together or glued together. And finally, we have our mature mRNA ready to leave the nucleus and go to the cytoplasm. So who was responsible for cutting out the introns and splicing together the exons? So this was done by a catalytic RNA called a ribozyme. And that ribozyme, with the help of a protein, together they carried out or catalyzed this splicing, RNA splicing process. So as mentioned earlier, spliceosomes are going to carry out RNA splicing. Spliceosomes consist of a variety of proteins and several small RNAs that recognize the splice sites. The RNAs of the spliceosome also catalyze the splicing reaction. These catalytic RNA are called ribozymes. So here is a spliceosome, and we have the small RNAs, which are also called the ribozymes. And the purple part is going to be the protein. So the spliceosome is going to cut out the introns and splice together or glue together the exons. Ribozymes are catalytic RNA molecules that function as enzymes and can splice RNA. The discovery of ribozymes rendered obsolete the belief that all biological catalysts were proteins. So before the discovery of ribozymes, everyone thought that enzymes were the only catalysts. 3 properties of RNA enable it to function as an enzyme. It can form a 3-dimensional structure because of its ability to base pair with itself. Second, some bases in RNA contain functional groups that may participate in catalysis. And third, RNA may hydrogen bond with other nucleic acid molecules. Some introns contain sequences that may regulate gene expression. Some genes can encode more than one kind of polypeptide, depending on which segments are treated as exons during splicing. This is called alternative RNA splicing. Consequently, the number of different proteins an organism can produce is much greater than its number of genes. Okay, so what does alternative RNA splicing allow? It allows one gene to produce more than one type of protein. Proteins often have a modular architecture consisting of discrete regions called domains. So basically, proteins just have different sections called domains. In many cases, different exons code for the different domains in a protein. Exon shuffling may result in the evolution of new proteins. So here we see the correspondence between exons and their protein domains. So we know that the exons are the coding regions of our gene. And each exon is going to correspond to a section of the protein, which is called the domain. So domain 1 corresponds to exon 1, domain 2 corresponds to exon 2, and domain 3 corresponds to exon 3. But this whole molecule right here is just one protein, or one polypeptide. Genetic information flows from mRNA to protein through the process of translation. So we are at our last step of gene expression, where mRNA is going to be used to synthesize a protein. A cell translates an mRNA message into a protein with the help of transfer RNA, also called tRNA. So what does transfer RNA transfer? Transfer RNA is going to transfer amino acids, which are the building blocks of protein or polypeptide. The tRNA is going to transfer amino acids to the ribosome, and the ribosome is going to help connect or bond the amino acids together into a chain, which is the polypeptide, also called the protein. Translation is a complex process in terms of its biochemistry and mechanics. Each tRNA molecule enables translation of a given mRNA codon into a certain amino acid. Each tRNA carries a specific amino acid on one end and has a specific anticodon on the other end. The anticodon base pairs with a complementary codon on mRNA. So basically, each tRNA is going to match with a group of 3 nucleotides on the mRNA called the codon. So here we see the tRNA with the amino acids on the top and the anticodon on the bottom. And the tRNA are going to help transfer the amino acids to the ribosome. Once the tRNA arrives at the ribosome is going to help elongate the chain of amino acids to form the polypeptide. And notice also how the ribosome is reading the mRNA, which is going to be used to bring in the transfer RNAs and build that polypeptide. A tRNA molecule consists of a single RNA strand that is only about 80 nucleotides long, flattened into one plane to reveal its base pairing. A tRNA molecule looks like a clover leaf. Because of hydrogen bonds, tRNA actually twists and folds into a 3-dimensional molecule. tRNA is roughly L-shaped with the 5' and 3' ends both located near one end of the structure. The protruding 3' end acts as an attachment site for an amino acid. So that's the top part. So here we have the attachment site of the amino acid at the 3' end. And we have the anticodon at the bottom of the tRNA. The anticodon is going to be composed of 3 nucleotides, which are going to match with the 3 nucleotides of the codon on the mRNA. We have 3 different pictures of the tRNA. The tRNA to the right is a simplistic picture of tRNA, with the amino acid attachment site at the top and the anticodon at the bottom. Accurate translation requires 2 steps. First, we need to connect an amino acid to tRNA. The enzyme that helps to catalyze that is aminoacyl tRNA synthetase. Second, a correct match between the tRNA anticodon and the mRNA codon has to take place. However, there is flexible pairing at the 3rd base of a codon. This is called the wobble position, and this allows some tRNAs to bind to more than one codon. So here we see a aminoacyl tRNA synthetase, in this case tyrosyl tRNA synthetase, is going to add the amino acid tyrosine to our tRNA right here. So once it does that, the aminoacyl tRNA is released from the enzyme. And notice how this process requires energy. Next, we will discuss the protein factory, which helps to build the polypeptide by connecting the amino acids together. Ribosomes facilitate specific coupling of tRNA anticodons with mRNA codons in protein synthesis. The two ribosomal subunits are called the large and small subunits, and they are made of proteins and ribosomal RNA, also called rRNA. Bacterial and eukaryotic ribosomes are somewhat similar but have significant differences. Some antibiotic drugs specifically target bacterial ribosomes without harming eukaryotic ribosomes. A ribosome has 3 binding sites for tRNA. Because ribosomes are going to receive the tRNA, which is going to transfer the amino acid to the ribosomes for it to create the polypeptide. The P site holds the tRNA that carries the growing polypeptide chain, hence it starts with the letter P. The A site holds the tRNA that carries the next amino acid to be added to the chain, and the E site is called the exit site. This is where the tRNA is going to leave the ribosome after it has transferred the amino acid. So again, here we see the ribosome, and we see that the ribosome has 2 subunits. The large subunit is at the top, and the small subunit is at the bottom. The large subunit has the binding sites for the tRNA, E, P, and A. And also notice right here, the small subunit is going to have the mRNA binding site. So the mRNA is going to attach to the small subunit, as we see here on the right. And each tRNA is going to complementarily match to each codon in the mRNA sequence. And we remember that the codon on the mRNA is going to match with the anticodon on the tRNA. There are 3 stages of translation, and actually they're going to have the same names as the 3 stages of transcription. We have, again, initiation, elongation, and termination. Initiation is going to start translation, termination is going to end translation. And in elongation, the amino acid chain is going to be elongated to form our polypeptide. All 3 stages require protein factors that aid in the translation process. Energy is also required for some steps too. In initiation of translation, the start codon is going to signal the start of translation. The start codon is going to be located on the mRNA. First, a small ribosomal subunit binds with mRNA at the mRNA binding site and a special initiator tRNA. Next, the small subunit is going to move along the mRNA until it reaches the start codon. Proteins called initiation factors bring in the large subunit that completes the translation initiation complex. So here we see how the mRNA is going to bind to the small subunit at the binding site, and the ribosome is going to slide until it reaches the start codon right here. And at the start codon, the initiator tRNA is going to match with the start codon with its anticodon, and it's going to bring in our starting amino acid, which is methionine, MET. And if we zoom in here with the anticodon on the tRNA and the codon on the mRNA, we can see how they match. So after that, the large subunit is going to be brought in and attached to the small subunit, and we can see how the initiator tRNA is bound to the P site of the large subunit. This process requires energy in the form of GTP. And now we have our whole initiation complex here complete. The next step of translation is elongation. This is where amino acids are going to be added one by one to the C-terminus of the growing chain. The C-terminus is referring to one side of the amino acid, the other side being the N-terminus. Each addition involves proteins called elongation factors. Elongation occurs in 3 steps: codon recognition by the anticodon, peptide bond formation connecting the 2 amino acids together, and translocation, moving the ribosome so that we can move the tRNAs to the next slot on the large subunit. Energy expenditure occurs in the first and third steps. Translation is going to proceed along the mRNA in a 5' to 3' direction. The ribosome and mRNA move relative to each other, codon by codon. The elongation cycles take less than 1/10 of a second in bacteria, so it happens really fast. So here we see a step-by-step diagram of elongation. So the first step is codon recognition. This is where the new tRNA is going to bind to the codon at the A site of the ribosome. And after doing so, the growing polypeptide chain on the tRNA at the P site is going to transfer to the amino acid at the A site. And a peptide bond is going to form, connecting the growing chain to the single amino acid at the A site. The next step is translocation. So notice how the P site tRNA doesn't have any amino acids. So in translocation, the ribosome is going to move or shift to the right, which causes the mRNA to shift to the left, along with the tRNAs. So the tRNAs move one slot to the left. The tRNA in the P site entered the E site, which is the exit site, which kicked out the empty tRNA. And also the tRNA at the A site moved to the P site with the long chain of amino acids here. Okay, so this cycle is going to repeat. A new tRNA is going to come in again at the A site, and so on. But when does translation stop? And that brings us to termination. So termination is going to happen once we reach a stop codon on the mRNA, which is a group of 3 nucleotides that code for a protein called a release factor. The release factor is going to result in the stopping of translation, and also it's going to disassemble the ribosome, detach the polypeptide, detach the mRNA. So basically it disassembles everything, our translation initiation complex. So here we see the diagram of the termination of translation. Once the ribosome reaches the stop codon on the mRNA, a release factor is going to enter the A site and cause the disassembly of the whole translation complex. The tRNA is going to be released, the polypeptide is released, the ribosome subunits disassemble, also the mRNA is released. Often translation is not sufficient to make a functional protein. Polypeptide chains are modified after translation or targeted to specific sites in the cell. So even after we make the polypeptide after translation, the polypeptide might need to be folded further and also further modified in order to produce our final protein product. During its synthesis, a polypeptide chain begins to coil and fold spontaneously into a specific 3-dimensional shape. So a 3-dimensional molecule with a secondary and tertiary structure. A gene determines primary structure, and primary structure in turn determines the shape. Post-translational modifications may be required before the protein can begin doing its particular job in the cell. And so post-translational just means after translation. and modifications mean changes in the polypeptide. Two populations of ribosomes are evident in cells. We have free ribosomes, which are located in the cytoplasm, and mostly free ribosomes synthesize proteins. Bound ribosomes are going to be attached to the rough endoplasmic reticulum, and they make proteins of the endomembrane system and proteins that are eventually secreted from the cell. Ribosomes are identical and can switch from free to bound. Polypeptide synthesis always begins in the cytosol. Synthesis finishes in the cytosol unless the polypeptide signals the ribosome to attach to the ER. Polypeptides destined for the ER or for secretion are marked by a signal peptide. And remember, ER means endoplasmic reticulum. A signal recognition particle, SRP, binds to the signal peptide. The SRP escorts the ribosome to a receptor protein built into the endoplasmic reticulum membrane. So here we see a diagram of SRP escorting a ribosome to the rough endoplasmic reticulum. So step one, we see that the polypeptide is being synthesized, or it's beginning. Step two, the SRP is going to bind to the signal peptide, which is part of the polypeptide chain, and that allows it to escort the ribosome to the endoplasmic reticulum, where the SRP is going to bind to the receptor on the endoplasmic reticulum, and the SRP is going to detach, and the polypeptide is going to continue being synthesized. The polypeptide goes inside of the lumen, or the empty space inside of the endoplasmic reticulum, and also the signal peptide is going to be cleaved off by an enzyme, and the polypeptide is going to fold into its final protein form inside of the rough endoplasmic reticulum. Multiple ribosomes can translate a single mRNA simultaneously, forming a polyribosome or polysome. This allows for quicker translation. So polyribosomes enable a cell to make many copies of a polypeptide very quickly. So here we see a polyribosome made up of a line or assembly of ribosomes, making the polypeptide much faster. A bacterial cell ensures a streamlined process by coupling transcription and translation. In this case, the newly made protein can quickly diffuse to its site of function. So earlier we mentioned that because bacteria do not have a nucleus, transcription and translation are not going to be separated. They both happen in the cytoplasm. Therefore, we can see here that transcription, which is DNA to RNA, and translation, which is RNA to a polypeptide, is happening simultaneously here. In eukaryotes, the nuclear envelope separates the processes of transcription and translation. RNA undergoes processing before leaving the nucleus. As we saw earlier, there are two things that happen in RNA processing: adding the 5' cap and the poly A tail to the ends, and also cutting out the introns and splicing together the exons. So here is just a whole summary diagram of gene expression inside of a eukaryotic cell. We have transcription, RNA processing happening inside of the nucleus, and we have translation happening in the cytoplasm at the ribosomes. The tRNA are going to transfer the amino acids to the ribosomes, and the ribosomes are going to connect the amino acids together to form our polypeptide chain.
In translation, a cell reads an mRNA message and assembles a polypeptide accordingly. We can divide translation, the process of building a polypeptide, into 3 stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anticodon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine. Proteins called initiation factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with the large ribosomal subunit to form an initiation complex. The initiator tRNA sits on the P site on the ribosome.
Next, we will talk about mutations. Mutations are changes in the genetic information of a cell. This is going to happen at the DNA level. Point mutations are changes in just one nucleotide pair of a gene. The change of a single nucleotide in a DNA template strand can lead to the production of an abnormal protein. If a mutation has an adverse effect on the phenotype of the organism, the condition is referred to as a genetic disorder or hereditary disease. Here's an example of a point mutation in the beta globin gene, which is going to code for the protein hemoglobin in your red blood cells, which carry oxygen. The normal beta globin gene is shown on the left, also called the wild type beta globin gene. So we can see right here, we have a CTC for the template strand, but for the mutated beta globin gene, we have a CAC instead. The T has been substituted for AA. This results also in a T instead of an A in our non-template strand. This is also going to result in a substitution of an A replaced with a U, which in the end is going to code for a completely different amino acid. Instead of having GLU, which is glutamic acid, we have VAL, valine. This is going to cause your red blood cells to not have a normal round shape, but instead a sickle shape due to a misshapen hemoglobin protein. Point mutations within a gene can be divided into 2 general categories. We have single nucleotide pair substitutions and nucleotide pair insertions or deletions. A nucleotide pair substitution is when you replace one nucleotide and its partner with another nucleotide and its partner. Kind of like replacing the original teacher with a substitute teacher. A silent mutation has no effect on the amino acid produced by a codon because of the redundancy in the genetic code. So for example, if we had the codon UAU and we mutated it to UAC, so we substituted U for a C, that would not change the amino acid that we would get, because both UAU and UAC, they both code for the amino acid TYR, which is tyrosine. A missense mutation still codes for an amino acid, but not the correct amino acid. So for example, if I had UAU and I substituted the A for a U, this would result in a different amino acid. So earlier we saw that UAU is going to code for TYR, tyrosine amino acid, but UUU is going to code for the amino acid phenylalanine, PHE. A nonsense mutation changes an amino acid codon into a stop codon. This most likely leads to a non-functional protein. So, for example, let's say that we had UCA, and we changed UCA into UGA right here. So UCA right here is going to code for the amino acid serine. However, when we substitute the C for a G, this changes the serine amino acid into a stop codon. Or this is a stop codon. So the UCA codon turns into a stop codon. And we remember that stop codons are going to code for a release factor, which is going to stop translation. Okay, so here we see some examples once again. We have a silent mutation. So if you look at the codon right here, CCA is going to transcribe into GGU. So we look at the DNA sequence at the top. This is the normal DNA. So if you go up right here, the original sequence is CCG. So we changed the G, we substituted the G for an A instead. But notice how the amino acid did not change. And that is because we changed the third position of the codon. So that's not really going to change the amino acid. It's going to be the same. There is a specific term for the third position of the codon. This is called the wobble position. So if you change the third nucleotide, it does not change the amino acid in the end. Insertions and deletions are additions or losses of nucleotide pairs in a gene. These mutations have a disastrous effect on the resulting protein more often than substitution mutations do. This is because insertion or deletion of nucleotides may alter the reading frame, producing a frame shift mutation. So this is going to change the order in which we read the groups of codons. Before we get to the examples of deletions and insertions, let's take a look at a missense mutation. So in the original DNA sequence, we have CCG as our second to last triplet. But as you notice right here, instead of having C as our first position, we change it or substitute it into a T. So that's not the third position, that's the first position. So as a result, instead of having the amino acid GLY, it's going to change into SER from glycine to serine. Spontaneous mutations can occur during errors in DNA replication, recombination, or repair. Mutagens are physical or chemical agents that can cause mutations. So these come from the environment. And spontaneous mutations come from within. Chemical mutagens fall into a variety of categories. Most carcinogens, which are called cancer-causing chemicals, are mutagens. And most mutagens are carcinogenic, causing cancer. The idea of the gene has evolved through the history of genetics. We have considered a gene as a discrete unit of inheritance, a region of specific nucleotide sequence in a chromosome, a DNA sequence that codes for a specific polypeptide chain. A gene can be defined as a region of DNA that can be expressed to produce a final functional product that is either a polypeptide or an RNA molecule. So this is an interesting point because sometimes we can just stop right at the RNA and not even translate it to a protein, and the RNA can be considered functional. So it has a certain job that it does. So let's take a look at examples of insertion or deletion mutations that result in frameshift mutations. This mutation is called a nonsense mutation. A nonsense mutation is going to result in producing a stop codon instead of a amino acid. So in our original sequence, we had an LYS for our second amino acid. But because we substituted the T for an A, this changed the amino acid from lysine to a stop codon. And therefore, this is called a nonsense mutation. We prematurely stopped translation. We have only one amino acid instead of the original 4 amino acids in the original polypeptide chain. Next, we have an example of an insertion mutation that leads to a frameshift mutation. So in our original DNA sequence, we had the triplets TAC and TTC and AAA, CCG, and ATT. However, in our insertion mutation, we add an extra A in between TAC and TTC, and that results in a frameshift, meaning instead of reading it as TAC and TTC, now we read the mutated DNA as TAC and ATT because of that extra A. Not only does it change the second triplet, but it changes the triplets down the line. So CAA, ACC, GAT. And in this case, not only is it a insertion and frameshift mutation, it also kind of indirectly resulted in a nonsense mutation because the second amino acid ended up becoming a stop codon. Here we have an example of a deletion mutation. So again, we have TAC, TTC, AAA, CCG, ATT. We have 5 triplets, but we're going to delete one nucleotide. We're going to delete the third A in the third triplet. So A is missing. And this is also going to cause a frameshift mutation because removing 1 is going to change the order. So we have TAC, TTC, AAC instead of AAA in the original. And we have CGA instead of CCG in the original. And instead of having ATT, we have it. We don't even have a triplet in the end. This is going to result in having a LEU and an ALA instead of a PHE and a GLY. So we can kind of see how insertions and deletions, they cause a frameshift mutation, which is going to have a more devastating effect or bigger change on the final amino acid sequence. Okay, so here we see an example of a deletion, but the deletion mutation does not lead to a frameshift mutation. Okay, And this is because it's not a single nucleotide deletion. It's a 3 nucleotide deletion. So in the original, again, we have TAC, TTC, AAA, CCG, and ATT. We have 5 triplets, but we're going to get rid of TTC, the second triplet right here. That's going to result in us only having TAC, AAA, CCG, and ATT. But notice how the remaining triplets, they're still going to be the same. We just have one triplet that's missing. So this would not be considered a frame shift mutation because we did not change the reading frame. However, we're going to be 1 amino acid short. So we had lysine in the original amino acid sequence, but right here we can see that it's missing because we don't have the codon that codes for it.